Genoa, do you have your slides ready? Fantastic. So our next speaker today is Dr. Genoa Warner, Assistant Professor at New Jersey Institute of Technology, and she is going to talk to us today about sex-specific effects of nanoplastic-induced placental toxicity. So Genoa, take it away.
All right. Hi, everybody. Thanks so much for having me. I'm really excited to tell you a little bit about my lab's work. I'm sitting outside right now because it's beautiful in New Jersey, but if hopefully the ambient noise won't be too loud, I can always duck inside if I need to. All right, so I'm going to start by telling you a little bit about the placenta because some of us might not be reproductive biologists. The placenta is a temporary organ that forms during gestation to support the fetus. It's the interface between the mother and the fetus. All the nutrients, wastes, etc., pass through the placenta on their way to and from the fetus. And it's got a pretty cool structure that allows for this exchange while keeping the fetal and the maternal blood separate from each other. As they make their way into on the fetal side, there's the umbilical cord. I've been studying the mouse as a model, so I need to have a little disclaimer, right? The mouse and the human placenta are not exactly the same, but they have the same function. And although some of the structures, especially have different names, it is even hard for me to keep straight sometimes, which bit corresponds to which bit. Um, Generally now when we're looking at the mouse placenta here, we have three main layers, and the maternal fetal exchange is primarily happening in this labyrinth zone, which corresponds to the chorionic villi, sort of central part of the human placenta. Now, I come originally from the world of endocrine disrupting chemicals. Phthalates, which are plasticizers, are my baby. I still study phthalates a lot. And there have been quite a few studies that have looked at the impact of endocrine disrupting chemicals on the placenta. So this figure here is from a review that looks at showing the different categories of endocrine disrupting chemicals connecting to different endpoints within the placenta that they've been shown to impact. We know a little bit about the impact of some types of nanoparticles on the placenta. This figure is a review from the Stapleton lab that was looking at metal nanoparticles, and it also does include some of the polystyrene nanoparticles here, finding impacts on the placenta related to apoptosis, inflammation, reactive oxygen species, etc. So the main goal of the work in my lab and the project that I'm going to tell you about now is to investigate how nanoplastics impact placenta development and function using a mouse model. And our biology hypothesis is that the nanoplastic exposure will impact essential signaling pathways, including hormone signaling. We've seen some preliminary evidence from this in other studies. But we also have a plastics related hypothesis too here, which is basically that not all plastics are the same. I'm sure we all know that the field has spent a lot of time studying polystyrene spheres and then has spent a lot of more time gnashing our teeth over how representative they are of every other type of plastic particle that's out there. So we are working towards sort of big picture wise, trying to look at a bunch of different particle types, because what we need is something like this figure, but for particles, for plastic particles, where I think we're going to be seeing some effects that are same and some effects that are different across particle types. And just like with phthalates, there are a lot of different types of phthalates. If we drill down into phthalates, they don't all have the same impact, um, as each other. And I think we're gonna be seeing something similar when we look at all different types of nanoplastics. So I'm just telling you about the first two types of plastics that we've looked at, but the goal is to look at a whole lot more. So our first study was our pilot study. This one we used polystyrene spheres. We actually did this experiment in early 2023. It was just after I was setting up my lab. It was my students' first attempt at breeding mice and dosing mice. It was fun. But we have now finally been able to analyze our results here. So what we did for this, initial pilot study was that we bought polystyrene, either 50 nanometer or 200 nanometer, and we picked some fluorescent ones so that we could try to visualize them. And we orally dosed pregnant mice from gestational day 8 to 15, which represents the period of time when the placenta is developing until it reaches full maturity. We didn't want to start too early and mess with implantation. We really wanted to focus on placenta development. So on day 15, we collected the placentas and the fetuses and maternal blood to analyze hormone levels, gene expression, and look at placenta morphology. I want to tell you a little bit about the dose that we picked. We just picked a dose because, I mean, it feels like we could pick almost any dose and justify it. But still, I want to tell you about like some of the context for the doses here. I'm sure we've all seen this image and heard this statistic, right? A credit card of plastic per week, which that comes from the upper end of this estimate, 5 grams per week. If we convert this estimate to an approximately 60 kilogram woman, that would be equivalent to zero point 0.2 to 12 milligrams per kilogram per day. So if we were to stop here, our dose falls somewhere in the middle of this estimate. And when we're studying phthalates, we usually use a human exposure estimate when we're dosing our mice. Um, but I've noticed that a lot of papers in the nanoplastics, micro nanoplastics literature have been using the mouse surface area conversion, which is a common thing in, pharmacology, toxicology, which basically adjusts for the fact that mice have a different surface area volume ratio. And if we do this standard calculation, then that would mean our equivalent dose to the credit card would be 2.5 to 145 milligrams per kilogram. So if we're thinking about it in this context, then our dose is on the low side. Now, something that I would really like to know to help us give our doses more context is, well, how much of this plastic is actually in the placenta? When we give a mouse X amount, how much can we detect in the placenta? And I don't have good numbers for you on that, but we're working on figuring that out with our mice. But that I would love to figure out what that number that is and then about calculate to what we think our dose should be, and then maybe relate that to what we know about plastics in human placentas. Anyways, we picked a dose of 5 milligrams per kilogram. We characterized the particles that we bought with our collaborator at NJIT. They measured the particle sizes for us. Turns out our 200 ones were actually more like 220 on average. The blue ones were 48 nanometers. So we did all the characterization to confirm that they were what we thought they were. We looked at their translocation into the placenta. These are some pictures taken by Phoebe Stapleton's lab to make sure that the particles were there. This was with the 200 nanometer particle. It is not quantitative, but it shows us there that they're there. So this was done using her Cytoviva microscope, which looks at a spectral library plot for any given polymer type and then matches the pixels of the slides to what the expected spectrum of the polymer is. B on this image is our control. We did not see any plastics there. C was our 200 nanometer exposed mouse where we saw plastics distributed in the placenta. I also sent some slides to a collaborator who's really good at fluorescent microscopy. I am not really good at fluorescent microscopy. And he took these nice pictures to show us that we can see the red and the blue particles in the placenta slides. These are representative images, but we are hoping to be able to compare the density of the plastics in the different parts of the placenta going forward. And then we looked at the morphology of the placenta. So I guess first we measured, placenta weight, fetus weight, and placenta efficiency, which is the ratio of the placenta weight to the fetus weight. And in this study, they were not impacted. And then one of my students furthermore measured the area of the different layers of the placenta. So if you remember our mouse placenta up here, we have three main layers, the decidua, the junctional zone, and the labyrinth. The bottom is what it looks like in an actual histology image. And what she did was we scanned all the slides and she traced the outline of each layer to calculate the area. And when we looked at the areas of the layers, we found that the size of the decidua was decreased in both of our treatment groups, and our 200 nanometer was statistically significantly decreased. We have a very small sample size here in our pilot study, so we're only able to detect very strong effects here. So the top graphs that we're looking at are just looking at all of the placentas. The dots are this is by dam. And then the bottom level here, we are looking at them based on sex. Oh yeah, and I forgot to copy. My key got cut off here. Um, the ones on the solid bars on the left are female. The bars with the lines across them are male. And so we see that there's a statistically significant decrease in decidua area in the males, um, at both, well, borderline for the 50, significant for the 200 treatment group, and that the effect on the male seems to be largely driving the effect that we see together. We also measured the size of the maternal blood spaces and the size of the fetal blood spaces. So these are, maternal blood cells here and fetal red blood cells. And then the intrahemal barrier, which is the distance between the maternal and the fetal blood spaces where things get exchanged. And in this case, we looked at them by dam and then broken down again by sex. And we did not see any effects on our blood spaces for this polystyrene study. We also looked at gene expression related to transforming growth factor beta signaling. We picked the signaling pathway because I had an ambitious student who wanted to come up with his own pathway to look at. This is an essential pathway in the placenta, controls a lot of different things. Um, and I put a little figure here showing the relationship between some of the factors. Basically, a ligand, for the example here is transforming growth factor beta 1, will interact with a receptor, and which will cause the phosphorylation of transcription factors from the SMAD family. These get phosphorylated and make their way into the nucleus to control gene expression. So our first level analysis was to look at the gene expression from some of these different transcription factors and ligands. And we found well, these are all borderline. So the caret means like p-value is less than 0.1. We were generous here because of our small sample size. Increases in the expression of SMAD2 and SMAD3, as well as GDF15. And when we break these down by sex, the SMAD3 effect was significant in the males at 200. And then we also had a borderline significant decrease in TGF-beta in the males at 50. So again, we're seeing male specific effects here. Okay, so we've mostly wrapped up that first study into a little paper, but we've been itching to move on to study some more interesting particles and do a better job where we have a bigger sample size. So our second dosing study, we use polyethylene terephthalate, or PET nanoplastics. These are the same particles that we've previously used to study in the ovary. And we are lucky to have a collaborator at NJIT who can grind these up for us. So he ground up some plastic bottles and characterized them. These are irregularly shaped particles. They're approximately 240 nanometers in diameter. And what we did in this study was we exposed mice from gestational day 7 to 15. We started using peanut butter dosing for this experiment, which is less stressful for the mice and also easier for the undergrads. In fact, I should mention that this entire PET project is run by two undergrads who are on T34 fellowships. So they dosed pregnant mice from day 7 to 15. And in this case, now we had 9 to 13 mice per treatment group. We did the same collection at the end, day 15, fetuses, placenta, blood. And then I want to mention what our treatment groups were. It wasn't just nanoplastics. We actually did a nanoplastic exposure and a phthalate mixture exposure. And I'm not going to tell you too much about the phthalate mixture because this is a talk about nanoplastics. But we were interested in the interaction between the two, as well as what each thing was individually doing. We analyzed placenta weights, fetal weights, and the fetus placenta ratio by sex, and we saw a almost significant trending decrease in the fetus placenta ratio in the males. Um, so that is where we're at right now. So to summarize, all of the plastic particles that we tested disrupted the placenta, but the effects were not the same, which is about what we expected. Um, but interestingly, what was the same was that both treatments had sex-specific alterations. So really important to look at sex differences here. For our polystyrene study, we found that exposure altered morphology and gene expression. The changes in the decidua we think are like that's pretty interesting. But we can't tell from our single time point if this is premature thinning, which is a thing the decidua does as the placenta matures. It reaches its maximum size right around day 15 when we collected, or if it never got as big as it should have in the first place. We can't really tell from that without doing some more time course studies. And our changes in expression of transcription factors suggest disruption of TGF-beta signaling, but we haven't done any measurement of phosphorylated versus unphosphorylated proteins, et cetera. So that's another next step. And so the last thing I'm going to leave you with is that we're thinking about how nanoplastics may be endocrine disruptors, and that's something that we'll be continuing to think about. I have a wonderful research group and collaborators who have helped me with all of this work. The polystyrene study was led by my first graduate student, Haneen, and the PET was led by my undergrads, Allison and Miriam, who are T34 fellows. They're doing amazing work. We have, thanks to all our collaborators who have helped with this project as well and the funding. All right, that's it for me.
All right, fantastic, Genoa. Really nice studies. If there are questions, please jump in or put them in the chat box. We do have time for some. I'll start. So when in your um, RNA-seq proportional analysis, you emphasized increased numbers of trophoblasts, but I was viewing it as a loss potentially of endothelial cells and mesenchymal cells. Have you looked at vascularization in these tissues to see if that might be altered?
Well, we haven't looked at anything else yet, but yes, right. What could be more of something else could be less of another thing. Um, yes, we would like to. Um, right now, all we've done with this is the RNA-seq so far
Okay.
with the data set. Yeah.
A loss of vascularization might be analogous to or promote gestational hypertension or preeclampsia later in delivery, so or later in term. So there might be something really worthwhile following up on.
Okay, I'm writing that down.
And there's a question from Kirene. Uh, what are the interventions or mitigations to reduce exposure to plastics in the placenta? Hmm.
Oh, you mean for pregnant humans?
Yeah. So sort of
Well,
the big question.
haha. Well, I guess I can speak to that as someone who has three children. Um, but yeah, I mean, reducing plastics in food contact, water contact are the things that I was mostly think about, especially including I'm always thinking about endocrine disrupting chemicals too, but including the sneaky things, right? Like aluminum cans that are lined in plastic that we might not think of as a source of plastic. But generally, I don't think we should be telling everybody that it's our problem when we're pregnant to avoid all the things, right? We should have a system where it's not our own personal responsibility. And I hope that we are working on getting there.
I really agree with that. I think there needs to be more attention at many levels of government to fix the problems we have and the stresses that mothers feel about this. That's cortisol. That's clearly impacting child development and maternal health. So I hate that that's the situation we're in right now, where it's thrown off on the individuals. Cheng Cheng?
Yes, that's a really nice work. And we seen like this sex-specific effect all the time related to cardiovascular health and metabolic health for the microplastic exposure studies. So your studies, the RNA-seq result are really striking. I'm curious, since you look at like fetus, right? Have you thought of the mechanism? What's, I know that this this take a study that take time and really complicated. But have you thought about why it only affect the male, not the female, and what's the potential like direction for the future mechanistic studies?
Yeah, mechanism is a good question here. And I spend a lot of time thinking about like nuclear receptor signaling in terms of endocrine disruptors, and that's not the kind of mechanism that we're talking about here.
Hmm.
I do think it's interesting to think about, just sort of like the energy balance is different in pregnancy in male and female placentas and
Hmm.
fetuses, right? That like
Yeah.
typically males will have larger placentas, if I remember right. Um,
Oh.
that there are sex specific differences in development aside from any types of exposures. That the males are a little more energy intensive.
Hmm.
The females can be a little bit more conservative. And so their responses to environmental exposures might be a little different in that context. And so we've been thinking about that, but again, like we haven't had a
Yeah.
chance
Yeah.
to look at that. But I want to look at energy balance. And I didn't mention that we also had some gene ontology terms related to like mitochondria.
Hmm.
And so looking into like some of those directions, that's another plan.
Yeah. Thank you.
All right. Well, we're close to time, and I'm not seeing other questions. If anybody has one, they can put their hand up and jump in. But otherwise, I want to thank Genoa for a fantastic talk, as well as Scott Coffin for his talk. This has been great. Um, I thank you both, and I hope you have a wonderful Friday and going into the weekend as well. That's it for us today. Thanks, everybody.
Thank you.
Thanks, everyone.
Thank you. Bye.